Ok, desculpa, eu estava atrasada aqui. Ana Paula, por gentileza, você pode começar?
Good afternoon, everyone. Now we are going to invite Dr. Flávio Lara from Oswaldo Cruz Foundation to give us his presentation. And I would like to thank you for accepting our invitation to participate in this course. It's a pleasure to have you here, and I hope that our students will have a lot of knowledge coming from you, and that they can have a lot of questions for you and to learn more of your work and what you have been doing all this time with a lot of collaboration. And thank you very much for this. And I would like to invite Karina Girard to present you to the students. Thank you.
Okay.
Hello, can everybody hear me?
Yes.
Yes. Good afternoon, everyone. My name is Karina Girardi. It's a pleasure to be here today to introduce the next speaker, Dr. Flávio Lara. Flávio is graduated in microbiology by the Federal University of Rio de Janeiro, and he has a master's degree in bioscience and biotechnology and a PhD in biochemistry. And he has been working as a researcher in public health at Oswaldo Cruz Institute since 2006, with the main focus on leprosy research. He's innovative, enthusiastic, and keeps in touch with different branches of leprosy research, and also is my PhD supervisor. So today he's going to talk about leprosy neuropathy with presentation entitled Central Metabolism, Modulation, and Leprosy Neuropathy. Thank you very much, Flávio.
Thank you, Karina, for your introduction. Thank you, Roberta and Ana Paula, for the invitation. It's nice to be here among all these very fancy and important researchers that are presenting the very nice class Monday and today. I will share my presentation. As Karina said, I'm interested in the whole of central metabolism on the as a molecular basis of leprosy neuropathy, and I came from a cellular microbiology laboratory hosted in Fundação Oswaldo Cruz, as Karina said, and my presentation is divided in 3 parts, and we can make 2 stops for questions after the first and the second part. So I will not lose our time talking about the importance to study leprosy nowadays. Here in Brazil, it's a huge problem of public health care. And the problem is that hemilipri is able to invade the Schwann cells, the cells that are involved in the synthesis of the myelin sheath, responsible to insufflate, isolate, and maintain the axon physiology of the peripheral nerves. And by doing that, hemilipri generates this neuropathy that's associated with the impair of motor neurons, sensitive and after motor neurons, that generate these unreversible neuronal lesions that we are interested to avoid. An interesting thing about neuronal lesion in leprosy is that the neural lesion persists even after the beginning of the treatment. So if we can use some combination of drugs together with the multiple chemotherapy, we can somehow just, if not revert, but at least stop the neural damage just at the beginning of the treatment. And that's one of the main topics of my work. as researchers, and that I will bring you to some data that to convince you that this is possible, and I will be talking almost focus on that topic the entire time. So let's first thing first. There are not a lot of work about that in the literature, but there are some very fine works about the molecular mechanism associated with leprosy neuropathy, and most of them are divided into different topics. The first one is the demyelination by differentiation of infected Schwann cells, and the other topic is the demyelination promoted by immune system recruitment. That's more related with Dr. Cressida on Monday. So, making a long story short, the Schwann cells as SOX10 signaling as the ID card. SOX10, what's the signaling that makes Schwann cells Schwann cells, make and produce myelin sheath and be attached to axons. And during infection, imilipri in green promotes this loss of signaling. The SOX10, that is this promoter that's translocated to the nuclei here, it's missing during the infection. And so these Schwann cells lost its identity, becomes confused and think that maybe it could be a fibroblast, for example, and lose all the myelin sheath and go away from the nerve to other region of the body. And more important, lose the protection of the axons, that is the physiological role of Schwann cells. And other problems that these are differentiated now, and they are able to recruit macrophage to the nerve because they start to produce lots of chemoattractants to macrophage. And as you can see in this time-lapse analysis, these infected Schwann cells are able to recruit this macrophage to the location. And then could be associated with the damage that Dr. Cressida showed to us, that infected macrophage are able to generate it in the nerve by nitrogen free radicals generation in the nerve. So what I would like to show here today, it's a different topic. It's a topic involved in central metabolism modulation during infection. The differentiation of infected Schwann cells could take about some weeks or months. The recruitment of immune system to infect nerves could take also several weeks or months or even years. But I would like to talk about modulation that could be made in minutes and sometimes in seconds. And to my point of view, are the first insult that the axon will suffer during infection of imilipri. So this is a representative figure of a neuron in scale. So if the cell body has this size, the axon length will take about 7,000 of loops like that. In the neuron, like in the neuron that it's located in the sciatic nerve, the cell body, it's about 1 meter of the axonal end that's connected in your foot. So it's not possible that this cell body control the protein synthesis that is located in this synapse. all the control of the nutrients, protein synthesis, lipid synthesis of this axon is made by the Schwann cells that are supporting this axon. So the Schwann cells is like a nurse. They need to be continually keeping the axon going and going. And that's a very nice draw about that. Unfortunately, it's not a Schwann cell, it's an oligodendrocyte. It's the central nerve system draw. But recently, Dr. Bogdan Beirovsky shows that all this signaling is also happening in the peripheral nerve system. So the important here is to know that everybody knows that neurons really enjoy glucose. The brain is the main sink of glucose in our body, but in fact, the glia cells are processing glucose to the neurons. The neurons receive lactate and pyruvate. And the glia cells are during the entire state to the axons through these transporters. There are specific transporters for the Schwann cells membrane and for the axon membrane related with this process. And by doing that, the Schwann cells keep the mitochondria in the axons going. And to produce the electron potential in the membrane, they take lots of energy, and this energy came from pyruvate and lactate. So another aspect, it's about this, that Emilipri is just in love with lipids. Emilipri really, it's addicted for lipids. In every cell that Emilipri entered, like macrophage, Schwann cells, adipocytes, fibroblasts, Emilipri increase the lipids content. The only exception that I saw until today is the adipocyte. Inside adipocytes, Emilipri decrease the lipid content, probably to increase the lipid content in other effect cells like Schwann cells. So this is a very classic work from our group, doing by Caterine Matos, describing that, in fact, Schwann cells increase the lipid content very fast. And Emilipri is just be rapid by these lipids and recruiting lipid buddies and be covered by it. And we hypothesize that this could be a nice way to avoid innate immune system, but also Emilipri depends on lipids for their metabolism, cholesterol, for example. And it's a very untypical case because Emilipri, during the adaptation to live inside our cells, he just give up of a huge number of genes, and among them genes involved in lipids catabolism. So as cholesterol, for example, Dr. Angela Marx described in her work that Emilipri is not able to use carbons from cholesterol, but still cholesterol is very important for Emilipri viability because Emilipri still keep one enzyme that's able to reduce, to oxidize cholesterol to cholesterol. So the relationship between Emilipri and lipids is still tricky. Emilipri is doing different things than TB do with lipids, not just degrading the lipids to use carbons, but also using lipids maybe for reduce power or to just be covered by lipids and somehow do not expose its own antigens to the whole cell. The fact is, one of the first projects that I have been involved in the laboratory was to use of statins to inhibit cholesterol synthesis in the whole cell. And we observed observed that by doing that, we decrease the viability of hemoliprin inside macrophage. Here we use low dose and high dose of atorvastatin and simvastatin and observe the reduction of hemoliprin viability. This is the gold standard, that's rifampicin in higher dose, and this is rifampicin in lower dose. And we can see that the combination of rifampicin lower dose with atorvastatin in lower dose is able to mimetize the high dose of rifampicin. Or in other words, the statins to potentialize in 10 times the action of rifampicin. So this could be good news, even to more important to TB because of the multi-drug resistant TB. And we can see here in TB, it's almost the same, also in macrophage. And unfortunately, this work was not transposed to patients until nowadays. We already draw a clinical trial, but doesn't work. And this is a little frustrating in the work with leprosy, that it depends on a very nice collaboration with physicians, and this is very hard to do. And we have this difficulty to transport the data that we generated in vitro to the patients. But we do that in vivo, using animals, and we observe that this dose of atorvastatin is able to reduce cholesterol levels in the serum of these animals, and also to reduce viability of hemoliprin, not the statins alone. You can see that statins alone, even reducing cholesterol, has no impact over hemoliprin viability. But combining statins with a low dose of rifampicin, we can achieve a response similar than the high dose of rifampicin. So it's able to potentialize the effect of rifampicin in vivo too. And more interesting, the animal that receive atorvastatin plus rifampicin present a very low inflammatory response in the tissue. So statins are also able to reduce, probably because of the inhibition of this enzyme, this step limits enzyme, MG-CoA reductase. You not impair only cholesterol synthesis, but also prenylated proteins not possible to be synthesized, because geranil is inhibited also. So receptors like TNF that depends on isoprenylation to be linked to membrane are impaired in the use of statins. So we believe that this is why this reduction in inflammation, in tissue inflammation. So this could be also a good side effect to control neural damage and also tissue damage in a reaction episode, for example. On the other side, statins have lots of collateral effects. Cholesterol is important to the regeneration of myelin sheath, so there are some issues that we need to be careful when translating this data to humans. But in patients with multi-resistant TB, for example, that they just the chance of cure is reduced, this could be a nice trial to be done. So as conclusion of this first part, we can say that cholesterol is important for hemoliprin infection, and statin presents a synergistic effect with rifampicin, and also that atorvastatin rifampicin association impairs hemoliprin growth and tissue damage in shepherd bulb C mice model. So let's make a small pause now. If you have questions for this part, I can answer now, and also drink a piece of water. No question at all?
If anyone wants to speak in Portuguese, you can ask directly to Dr. Flávio, please.
So, if there is no questions, I can go to the second part. And maybe if you have question for this part, we can answer later. There's no problem. So, the hypothesis of the second part of my presentation is about if this increase in lipid synthesis and lipid scavenger by the host cell could, mainly the Schwann cell, may somehow impair axonal physiology. And we think that because there is vast literature showing that lipids are neurotoxic. And the most beautiful work about this subject was made by Jeffrey Milbrand group, showing that this is a conditional knockout mice that after induction by drugs, this animal get knocked out by a protein that's very important for the replication of mitochondrial DNA. So these animals lost mitochondrial in Schwann cells after 1 month of life. And after 2 months, they just drop the number of myelinated axons. And they do that not by losing Schwann cells, but by losing axons, by losing neurons. So the mitochondria in the Schwann cells are important to the physiology of the axons. And the problem is that these Schwann cells that lost the mitochondria increase their content of lipids. He represented as acylcarnitine. And they also observed that lots of other different kinds of fatty acid, long-chain fatty acid are enriched in the Schwann cells of this animal. And it's known by a long time that these long-chain fatty acids disturb the electron potential in the membrane of the axons. So we start at the beginning observing the glucose that most of the de novo lipid synthesis in our body are made by glucose, and we start to observe the glucose uptake of the cells. Here we use a fluorescent glucose analog, and we observe that in fact Schwann cells increase the glucose uptake, almost double the glucose uptake. And this was also observed by the group of Dr. Beirovsky, that released this year an article observing that after a lesion in the nerve, the Schwann cells increase most of the virtually all enzymes in the glycolysis. This is a response to damage. The Schwann cells increasing glucose uptake. And we are wondering what's the beneficial of emiliprid to induce that. I believe that most of things that host cell do during infection is from the best interest into emiliprid. Emiliprid just maneuver the host cell to do all the things that he wants that they do. So we observe that this increase in glucose uptake, it's a dose dependent of infection. If you increase the MOI of the infection, we have increase in the glucose uptake. Dead irradiated emiliprid generated in less intensity this increase of glucose uptake. BCG is not able to do that, and probably emiliprid is doing that by increase GLUT1 receptor. That's the main receptor of glucose in our body. And probably this is related with EGF-1 signaling that that our group also demonstrated in the past. So what's the thing about the increase of glucose and glucolysis in the host cell? In this experiment, we use isotopic labeling glucose in this 6 carbon, and we give this to the cells. In this experiment, macrophage infected with M. tuberculosis, and observe the enrichment of isotopic labeling carbons in the amino acids. And we observe here that the tuberculosis presents the same profile of enrichment than the host cell. So M. tuberculosis inside macrophage is just stealing amino acids from the macrophage. But emilipri do the opposite. Emilipri synthesize their own amino acid and generated different labeling amino acid from the host cell. And he doing that by using glucose or some glucose intermediary. So emilipri is stealing glucose from the host cell. And he's for sure being benefited by the increase of glucose uptake during infection. And another feature during this glucose uptake is the reduction of mitochondrial respiration. Here we observe the fluorescence of respirating mitochondria. This mitochondria has electropotential. They are producing ATP, so they are consuming pyruvate and generating CO2, water, and ATP. And during infection, we just drop this production to below our sensitivity method here. We can observe a huge drop in the mitochondrial electropotential. We observe this data in FACS analysis and observe that dead irradiated emilipri is not able to do that, to reduce mitochondrial respiration, as neither BCG are able to do that. And the respiration, the oxygen consumption of these infected Schwann cells are impaired. Most important, if you use antimycin, that's respiratory poison, is an inhibitor of the complex 3 from the respiratory chain in the mitochondria, we kill virtually 100% of Schwann cells. But if these Schwann cells was infected 2 days earlier than the contact with antimycin, we observe non-death. There is no death at all of these cells. All dead cells are staying here with propydium iodide, and this culture, this infected culture, is just insensible to this poison because these cells are not respirating anymore. These cells are fermentating glucose. And by doing that, by lose this electropotential in the mitochondria, emilipri just not alter the ATP synthesis in their host cell, but also alter the retrograde signaling, like mitochondrial host, that is pretty much important in the macrophage differentiation and activation of inflammation, for example. We observe that macrophage present this kind of reduction in the electropotential too. But more important of this mitochondria which lose potential recruit ubiquitination by paracrine and be directed to mitophagy. And that's why after 3 days of infection in vitro, we observe a drop in the mitochondrial DNA in these cells. And also we observe a drop in mitochondrial DNA in the nerve of patients suffering from leprosy neuropathy, in contrast with patients suffering from other different neuropathy not related with leprosy. This represents a reduction in the mitochondrial content in the nerves. And we believe that this happened because during all these modifications, after entering the cell, Emilipri activate all these genes involved in cholesterol biosynthesis, lipid de novo synthesis, and increase lipid bodies, also increase the expression of receptors involved in the uptake of cholesterol, increase the receptor of involved in the uptake of glucose, and activated de novo lipid synthesis to generate this intracellular heaven to Emilipri, that is this lipid sea that he can swim in that. And as collateral side effect, increasing the content in the cytosol of free fatty acids, Emilipri is able to open this electron transition pore in the mitochondria. This is a very very well described process that free fatty acids are able to open this transition pore and depolarize mitochondria, directing mitochondria to the mitophagy. And by doing that, mitochondria is another bacteria pretty much more adapted to live inside our cell than Emilipri. But Emilipri see in mitochondria competitor carbons. And also by doing that, by directing mitochondria to mitophagy, Emilipri avoid beta oxidation of lipids, so increasing the lipid content, and also avoid the cytochrome C release signal apoptosis formation. So avoid the signaling of apoptosis generated by this organelle. Also avoid the activation of inflammasome, and also avoid the production, the consumption of pyruvate by mitochondria and the generation of very not interesting molecules to Emilipri like taconate. Taconate is a respiratory poisoning for Emilipri and for other bacteria. So we believe that this is a very conserved process to avoid innate immune system and to avoid apoptosis of the whole cell. But by doing that, by address mitochondria to mitophagy, Emilipri has the whole cell has to solve this problem in the generation of ATP. Fermentation is less effective in the production of ATP compared with the respiration. So by keep the same amount of ATP in the whole cell, adaptation is the increase of glucose. So the whole cell increase glucose uptake in order to keep the same amount of ATP. But the cells need to produce lactate, because like any other cells that choose this pathway, like cancers, they need to generate lactate to regenerate the NAD molecules, because the other way is just stopped. So if you don't generate the lactate, you will be without this NAD that is crucial for the keep the glucose going. So that's why they generated all these cells start this Warburg effect and generated lots of amounts of lactate. So we will check the lactate generation remember that neurons depends on lactate to survive. So you check the lactate and we get a little disappointed to observe that, in fact, Schwann cells actually drop the lactate release in the medium instead of increase like we expect. And this is very odd. Again, irradiated dead Emilipri is not able to do that, and BCG neither. And we also observe a drop in lactate serum, circulate lactate in the multibacillary patients compared with postbacillary ones. So, and this is a problem. We believe that this is one of the problems that we observe in the leprosy neuropathy. If Schwann cells are not able to transfer lactate to the neurons, these neurons will starve and suffer. And in the last instance, they will lose function. This process is kept by these transporters, and these transporters are sensible. The promoter region of the genes are sensible to lactate. If lactate drop, these genes increase to increase the transport of the lactate with rest. And we observe that these genes is pretty much upregulated during infection. That's another proof that the lactate levels in the cytosol is dropped. And we also observe that these transporters are increasing the nerve of patients suffering from leprosy neuropathy, indicating that lactate is down also in the nerve of the patients. So this is a problem, and this is probably related with the enzyme that convert pyruvate to lactate and lactate to pyruvate. It's the same enzyme. It's lactate dehydrogenase complex. It's a complex of 4 subunits, and these subunits could be like these H subunits involved in convert lactate to pyruvate, or these M subunits involved in convert pyruvate to lactate. And as you can imagine, Schwann cells are more rich in M subunits, and neurons present more H subunits. So we measure the expression of these M subunits in fact Schwann cells and observe that it's dropped. So infection reduce these M subunits, and actually also the infection do not alter the expression of the H subunits. That's why you also someone is with the open microphone, and we also observe that this subunit, this M subunit involved in the generation of lactate is reduced also in the nerve of the patients. So that's why by modulating the expression of this gene that Emilipri control the reduction of lactate. And what happened with this lactate? Why Emilipri it's working to keep the lactate inside whole cell? So we give radioactive lactate to the whole cell and observe that these cells produce radioactivity lipids. So this isotopic labeling lactate, these carbons of lactate is going to lipids, and they produce most of the carbons is going to fatty acid and deacylglycerol, but also we observe in the supernatant of this culture lots of other lipids stained with these carbons came from this lactate. So the lactate that should go to feed neurons are actually going to synthesize lipids in Schwann cells, and it's a combination of problems. There are two problems here. So we observe that to potentialize the lipid synthesis, the host cell needs to increase the pentose phosphate pathway. That's the pathway that will generate the NADPGH reduction power to synthesize lipids. And this pathway is coordinated by this step limit enzyme, the glucose 6-phosphate dehydrogenase. This enzyme that diverts glucose 6-phosphate from glucolysis to the pentose phosphate pathway. And we go to check the expression of this enzyme during infection, and we observe that the expression is pretty much high in infected cells. So the pentose phosphate pathway is also activated during infection. The activity of the enzyme is increased. And also we observe the expression is higher in skin lesion of multibacillary patients, demonstrating that this signaling is conserved also in macrophage, because in this lesion, most of the cells are macrophage instead of Schwann cells that's here in the neurons. That is also increase the expression of this enzyme. So the pentose phosphate pathway is increased in Schwann cells infected by imilipri, and this was also confirmed by this tool using isotopic labeling glucose. We observed that imilipri is able to synthesize phenylalanine inside the host cell. This is an essential amino acid. Our cells are not able to synthesize it, but imilipri is doing that using corismate. That is another indication that most of the carbon in the glucose is deviated to erythrose 4-phosphate and going to corismate. Almost 6 carbons of the phenylalanine are staining. In other words, came from glucose. So the pentose phosphate pathway is activated. And as conclusion of the second part, we describe this deviation of the glucose from the glucolysis to the pentose phosphate pathway, generating lots of reducing power that is using to produce lipid synthesis. And this lipid synthesis uses as carbons pyruvate that's not going to lactate and is deviated to lipid synthesis. And this is a problem to axons, because without lactate, probably their mitochondria are not producing energy, and so they will swelling. This is a very well recognized effect that mitochondria produce if it's not respirating, if it's not with electropotential, the swelling of mitochondria. And also we conclude that from the pentose phosphate pathway, imilipri is stealing erythrose 4-phosphate to produce phenylalanine, and also phosphoenol pyruvate and corismate. And also for sure, the lipids from the de novo lipid synthesis. And this is probably a very bad news to axons, because they are lack for the main carbon source, carbon and energy source, and also they will be covered by lipids that will impair the sodium-potassium transporter. So I will make another small pause here. If you have questions about
Flávio,
or the first.
There are some questions. Ximena can start with the first one, and then I'm going to read on the chat. So Ximena.
Yes.
Yeah.
Hello, Flávio. A great talk. And I feel like going back to high school, and hello. Yes.
I'm listening.
Okay. Okay. Thank you
Hello.
for a great talk on. I felt like going back to high school and and med school with all biochemistry.
Sorry about that.
And no, it was actually great, and actually many new things have come out since, and which is good as well. It has evolved. a lot more is known on metabolism nowadays. And I was actually raised the hand in your first part, but it took a time until my raised hand was seen. So actually was regarding then the first part that I had a question is, according to these findings of the effect of atorvastatin and rifampicin, the combination, lowering the dose, the killing dose of rifampicin or M. leprae for mycobacterium, would you recommend it to be used then together with multi-drug therapy? And would you think it would favor then the use of UMDT, like a reduction, which means a reduction of the 12 doses back to 6 doses for multibacillary, for the multibacillary scheme?
Well, no, if I draw a clinical trial using that, I will not alter the dose of rifampicin. I will keep the same dose. But before that, we can, because statins are very common used. So we can, at the first time, just observe if patients that use statins, they are already using statins when they start the treatment, if they recover better, if they produce less reaction, they suffer less reaction during the treatments, if they suffer neuronal impair reduction during treatment, we can use just observing this patient that's already using statins. And I talk with some physicians, and lots of them say me to, if the patient enter the multi-drug therapy using statins, they recommend to stop to use statins, because statins could be hepatotoxic in some concentrations. But
there is also a problem with neuropathy with statins.
Yeah, but for sure, you can monitor this hepatic enzyme, the transaminases, and observe if there is some damage or not. But I think that the first step could be just watch the patients that's already using statins when they start the multi-drug therapy, and check if this patient has a better response to the treatment. Could be a nice start.
Yeah, because we're developing that study at the outpatient clinic but after MDT, the end of MDT. So it would be like a booster in the lipid metabolism to rapidly reduce the bacilli, which is still take a long time to reduce. The bacillary load in the patient takes quite a long time, multi-drug therapy. So in a sense, we would expect that it aids that reduction, but it would probably be better if given together with rifampicin then while on MDT and not after MDT.
You mind if I jump in?
This study was interesting to observe if this less reactional episodes. But it's a very nice. I would like to take a look in the data if you already have some data about it.
Not yet. Not yet. No, just
very
jump
few
in
patients still. Very few patients, not enough.
Yeah, just recruiting now, and well, it just started, but just to clarify a little bit, it's important to mention that we are trying to obviously do first a safety analysis, right? So among those patients, we decided that would be better to just finish MDT prior to beginning of statins. And the other thing is that actually we try to do what you mentioned, Flavio. We look at our database to see the number of patients that are taking statins at intake, right? When they were diagnosed, and the number was not that high among multibacillary patients. So it would be very difficult to run a careful analysis with those primary outcomes that we are discussing, right? So it's a kind of more straightforward type of answer that what we are looking at now. So or what we are looking for now, which means that we going to do, do the statins for 1 year and see what happens with those patients in a very controlled environment, right? Some are taking, some are not. We are including concerning the BI of those patients and following them up to see the impact of all of those secondary outcomes that you mentioned, reactions and other than the BI. So let's see what's going to happen in the next.
I'm a little disappointed about the data in TB. We start a huge effort in UERJ with TB, but just doesn't work. And we also make a conversation with Margarete Glaucomo to do that with multi-drug resistant TB, but also doesn't go forward. But the idea is sometime maybe with this, when you take out this data in Lipri, maybe the TB people start to be more interested to check that.
So Flávio, there are some questions in the chat.
Retrospective is really hard because of the uneven. There are too many statins. There are different dosages, and all that. It gives too many variables that they're very confounding. Thanks a lot, Flavio.
Thank you, Ximena.
So the first question on the chat is about the lipid raft and the neuron. So question, Flávio, Dr. Flávio, the microdomains called lipid raft present in the plasma membrane of the cell may be related in the neuropathogenesis in leprosy because these microdomains are rich in cholesterol.
Yeah, for sure. I have some data about that. The third part of the presentation is entirely about that. You see that the lipids are involved in the impairment of neural function, and we are. I will show that in the third part, and we can return to this question at the end.
And a question from Bruno. If Schwann cells are great hosts for MLAP persistence and survival, is it possible to observe MLAP division in Schwann cells over time, like weeks or months in vitro? Are you aware of any study that have tested this idea before?
This is strange. Namely, inside Schwann cells are able to even synthesize a complex amino acid like phenylalanine, but we do not succeed in observe lipid diversion in Schwann cells, even primary ones. The Schwann cell that used in the study is a Schwannome. It's a cell lineage that grow very fast, so it's not possible to observe a bacteria that grow very slowly in a cell that grow very fast. but primary Schwann cells are able to stay there. If you just take out the mitogens, the Schwann cells, the primary will stay there, but still they, after 1 month, they start to be ugly and the culture goes bad, and we try it. This is one of my first things to do in the lab, and we spend lots of time in that, but Schwann cells are not a good model to grow in millipede.
Yes, and Ana Paula Junqueira asked, do you know any idea what proteins or subproducts are being produced by amilepri to modulate lipids or mitochondria? Is it any role for ABC transporters?
Sorry, can you repeat the question?
Do you have any idea what proteins or subproducts are being produced by amilepri to modulate lipids or mitochondria? Is it any role for ABC transporters?
That's a nice question. I think that Milton, it's interesting that observing microRNA of amilepri controlling expression of host cell. I'm more interested to observe ubiquitination of host proteins during infection. Parkin is one of most important genes that depends of the SNPs in there, could address, could be related with the susceptibility of the disease. So I think that ubiquitination is for sure important to the success of infection, but for sure, amilepri it's able to generate lots of microRNAs that will control the expression of host proteins, and I'm pretty much sure, and also Milton, I think, that this is one of the tools that amilepri use to subvert the whole cell to its own benefit. But again, related with ABC transporter, I never checked that, but could be interesting. There are lots of drugs that interfere with ABC transporter, and for sure could be a hole in that in the cytopathology. I never checked it.
Bruno Mieto asked if the glucose pentose phosphate pathway modulation seen in infected Schwann cells, do you think that this metabolic change starts as soon as amileptin contact the Schwann cell membrane, or only when the bacilli enter the host?
Well, we check this alteration in 48 hours after infection, and this is a more difficult task, and it's hard to make a time course, but the glucose uptake time course we made, and at 24 hours, it's not in the peak. The peak of the glucose uptake, it's about 48 hours. So I think that's a process that depends of the entry of amilepri, the activation of a series of genes involved in these transports, and after that, the effect. So it's like a fake propaganda that I make at the beginning that this alteration take about seconds. Most of them probably will take some hours to happen, because depends of the modulation of the expression of enzymes of the host.
So the last question. Marcia asked, have you seen anything about AMPK levels? This is an important energy sensor and regulates many cellular processes, including autophagy, and is described an important protein in glucose uptake and in regulation of these lactate transporters. How is working the TCA cycle?
AMPK, HIF1 alpha, it's an emitter, are the three complex that control the entire anabolism, catabolism in the whole cell. I would love to check that, but my PhD students, Karina, have lots of things to do, and we still don't have time to check that. I bought the antibodies in the past, but we still do not make that. But for sure, I think that will be a mix of signaling, because we see a signal of MPK and will not generate lipids, and the lipid generate lipids. You see the signaling of mTOR and will not increase the uptake of glucose so much as amylipid does. So I think that's a combination of these pathways. We have me, Bruno, and Bogdan Berovsky, we are discussing about that at about a couple of years, if I remember correct. And we already tried to send samples to check that in laboratory, but we just lost the samples in the transportation. But we will do that for sure. We need to check the mTOR, the HIF1 alpha, MPK signaling state in these cells for sure. But we still don't.
So that's all.
Okay, so I will go now to the last part of the presentation. I hope that everybody is still there. Okay. In the second part, we will check if that assumption is true. If in this situation, the lack of lactate transport to the axon will generate this cytotoxic effect in the axon. So these micrographies made in the electron microscope by Sérgio Antunes, and Bruno Mieto recovered these negatives, and we make this analysis. So we observe mitochondria here in the Schwann cells from nerves from patients suffering from other neuropathies, not related to leprosy. And we observe normal mitochondria there in the Schwann cells, normal mitochondria in the axons, unmyelinated axons, and also normal mitochondria in myelinated axons, okay? Mitochondria are very electron-dense structures in the electron microscope because they have lots of cytochromes that has lots of iron. So it's very easy to identify mitochondria in the electron microscope. And we also observe mitochondria in the Schwann cells of the patients, but we observe mitochondrial swelling in the axons of unmyelinated and myelinated nerves. So it's a little frustrating to observe that mitochondria in the Schwann cells are not alterated, but probably because these Schwann cells are not infected, but these axons has maybe lots of Schwann cells infected upward and inward, this image. So I think that, but the fact is, in these nerves from the leprosy patients are altered. It has increase in diameters that's showing that they are not respirating. And that's the famous picture that Dr. Cressida say that help her to understand her model in zebrafish. So we observe that so the nerve in the patients the mitochondria are suffering, and probably this happened first because you see a very nice myelinated axon there, and the mitochondria are completely altered. So even with this nice myelination, this axon is not able to synthesize, to produce energy and to conduct electron potential as well as this one, for example. So now it's also true that the unmyelinated axons in the leprosy patients presents a less mitochondrial number compared with non-leprosy axons. So the mitochondria is not only suffering from the lack of lactate, but it's also going to mitophagy, as we observe by the quantification of mitochondrial DNA. And this is probably a combination of factors, not only the harvest, but also the oxidative stress made by in fact, macrophage, as demonstrated by Dr. Cressida. So for sure, it's a very gray area with lots of signaling, lots of damage process, and we are characterizing this new one. So there is other aspects about the whole cell metabolism modulation during hemilipid infection, and one of that most interesting in my point of view is the increase in the reduction power synthesis. Hemilipid increase both sides in the generation of NADPH reduction generation. So malate enzyme is another huge source of reducing power, and the malate enzyme is also more expressed during infection and more active. And also it's more expressed in nerve of our patients. So it's the both sides of the source of NADPH are increased, and this is for sure pumping the fatty acid synthesis. But on the other side, another sink of NADPH is the glutathione system. Glutathione is one of the most important antioxidant pathway that we ourselves use. So it's also rely in the reducing power of NADPH, and glutathione, it reduces glutathione, stabilize free radicals being oxidized and be regenerated by the reducing power of NADPH. And this process is made by this enzyme, glutathione reductase, and we check the expression of this enzyme in these infected cells, and is pretty much increased during infection. So this process could be also be benefited by the increase of this NADPH. And the expression is also observed increased in the skin lesion, where it's reached in macrophage, and also in nerve lesions, in nerve biopsy, sorry, of our patients, compared with other neuropathies, nerves. So, more important, hemilipid is not only not generating oxidative stress in the whole cell. Here we are observing Schwann cells, but also if we challenge these cells with hydrogen peroxide, the infected Schwann cells reduce. They are more protected to oxidative stress than the control ones. Hemilipid is protecting its home from oxidative stress came from outside, and this protection it's based in the generation, the reduction potential generated by the pentose phosphate pathway, because if we use 6-NAN, that's an inhibitor of the pentose phosphate pathway, this protection, this difference between the infection and control, it's vanished. And also this difference is vanished if we use BSO, that's an inhibitor of the synthesis of glutathione. So this protection is dependent of glutathione, that is dependent of the pentose phosphate pathway. And that's why it's important to the hemiliprid to increase the pentose phosphate pathway of the whole cell. And maybe being prepared for this kind of insult that in fact macrophage could generate it in the nerves. But for sure, this is a balance of things, and with time, this process will win and the nerve will be degenerated. So returning to this draw that I showed you at the beginning, we now know that in fact, glial cell, in the case of hemiliprid, is reducing lactate transport to the axon. So there is no mitochondrial activity in this axon, and this is probably related with the loss of potential of action of these neurons. And also, this pyruvate is being diverted to the lipid synthesis. So we now would like to try to avoid this process, giving lactate to the neurons or impairing this division, this deviation of the carbons from lactate to lipids by inhibition of the pentose phosphate pathway. Because as you remember, the pentose phosphate pathway is important to generate the reducing power that will be used in the lipid synthesis. And if you inhibit the pentose phosphate pathway, you reduce the lipid synthesis. And we observe that inhibiting the pentose phosphate pathway, we recover the lactate released by the Schwann cell, even infected ones. And also we recover the mitochondrial loss of electropotential because we reduce the lipid synthesis. And also we observe that if we silence the sign of the pentose phosphate pathway. So now we have that's a very interesting tissue that it's rich in neurons cell bodies, and these neuron cell bodies is innervating the peripheral nerve system, and we can take out this nerve for sure, not from humans but from mice. This idea came from Dr. Bruno Mieto and is developed in my group by my PhD student Karina Girardi. And we had here this dorsal ganglia. In red, we can see this neuron cell body, and all these in green are axons that this neuron grow in vitro. So it's a very nice model. You can dissociate these neurons from this body, and they grow in vitro and make all the arborization. They are happy. In our model, we give this supernatant of Schwann cells to this neuron, and they became very happy. But if you give supernatant, and now returning to the question that I received during the pause, if we give supernatant of infect Schwann cells, these neurons, so if we infect Schwann cells but inhibit the pentose phosphate pathway using 6-NAN, we kind of recover these neurons. They are not so happy, but also they increase X, but it's some way a start. So this is a good example of the host target therapy that I was talking about. That's one of the main goal of my group. that's find some pathways of the whole cell that we can maneuver to avoid tissue damage. And this is one of them. Maybe we can avoid neuropathy by avoiding this kind of signaling that imilipri is imposing to the Schwann cells. But we to make two steps up front, we need to make one step back, and we use this Schwann model that's very easy and reliable model to make lots of analysis, and now we can measure viability of neurons. Just always giving supernatant of control Schwann cells or infect Schwann cells or infect Schwann cells plus lactate, and we can avoid the death of these neurons. And we also can avoid this neuron death by silencing the enzyme that's important to the pentose phosphate pathway, or by killing imilipri with rifampicin in the Schwann cell. All these things are happen in the Schwann cells, and we are giving the supernatant to the neurons and observing the viability. This is our model. And we observe that returning to the neurons from the dorsal roof ganglia, we can observe that adding lactate in the supernatant just partially recover the physiology of these neurons, but inhibit the pentose phosphate pathway, generate a better recovery. But still, we are talking about neurons with branching. But if you analyze the length of this branching, the 6-ANAN is pretty much better than the lactate. And also, if we analyze the branch complexity, we understand that we are still have a long way to go until we can claim that we have a drug that's able to avoid leprosy neuropathy. But with this model, we have the intention to try a group of molecules that we have in mind. So this indicated that it's not only the lack of lactate, but there is something else that's making damage to these neurons. That the 6-ANAN is pretty much avoiding. And we think in lipid that the Eccles phase of the supernatant is not neurotoxic at all. This is the supernatant of infected Schwann cells, and this is the Eccles phase. In the other words, proteins and carbohydrates that is in this supernatant. But the organic phase, the lipids of the supernatant that process this neurotoxic effect. So the Schwann cells, as I showed before, are not only consuming lactate, it's also converting this lactate to lipids, and these lipids is making damage to the neurons. And we also observe that PGR1 is the trigger. It's also fault of PGR1. So PGR1 is able to, the recombinant BCG expressing PGR1 is able to reduce lactate in the whole cell, and also beads covered by PGR1 exposed to Schwann cells are able to produce supernatant that is also neurotoxic. So PGR1 is involved with this entire signaling, and now that we have the one candidate that's 6-ANAN, we need an in vivo model, and this in vivo model we are drawing in the last couple of years in Instituto Lauro de Souza Lima in Bauru. We are using these nano-news to infect nerve of nude mice, and we are able to, I say we, but actually Bruno and Karina and Thiago is doing that, and Renata and Patricia, because I'm just staying in my table, writing emails, and from to, and he was able, using this needle, to inject. We are using beads, but he's able to inject these beads in the nerve without making any kind of physical damage in the nerve. That's very important to our analysis, because we need to observe the damage doing by hemiliprin and not by physical damage that will for sure recruit immune system and make lots of damage in the tissue. So this model it's still being developing in the Lauro Souza Lima. We can make very nice images from these nerves. And in this first analysis, you can still see some hemiliprin there, and some areas with this kind of loss of structure in the nerve. But this damage is pretty much visible after 28 days of infection, when we can see that this is probably hemiliprin side macrophage and making these bubbles of myelin that Dr. Cressida also observed in the zebrafish. So we have this model, and we will try a complex of targets in this model. We have inhibitors of polyunsaturated fatty acid, for example, ibuprofen, diclofenac, xyleuton. So acetyl salicylic acid. We are talking about 6-ANAN, that's an inhibitor of pentose phosphate pathway. That's a pathway that's very important to the control of ROS, as example, the oxidative stress. But we can also maybe control the leprosy neuropathy using aspirin. So this is just one example of how far we can go with these host target therapies. And also, this came from Dr. Cristiana Macedo, and these ideas came from Dr. Marcia Berredo. That's also, we would also check some cholinergic signaling inhibitors in these models. So, at the end, I would like to show some another diversion that came from this data. We use this increase in glucose uptake observing infectious one cells to try to give to our patients fluorodeoxyglucose. Is a isotopic labeling glucose analog that we inject in one of patients. Actually, we have now at about 4 patients, and we made it the first whole body image of the nerve infection in a patient. And we can see that this is a sciatic nerve. We can see the staining here of glucose that going until the foot of the patient. We observe very interesting, this data was generated by Dr. Sérgio in Federal University of Rio de Janeiro, and he had some very nice interpretation of this analysis. We observed staining in the heart, staining in the liver. We can observe nerve infection in places that we will never imagine before. And this is a very exciting tool that we are developing in collaboration with the Souza Araújo outpatients and Federal University of Rio de Janeiro. So this is just one example of kind of tools that could be generated by this data. And with this image, I make the conclusion of the part 3, that is hemiliprin deviated Schwann cells carbons from glycolysis to pentose phosphate pathway and from lactate to lipids. This metabolic modulation is crucial for the success of infection and could be related with axonal loss of function in leprosy neuropathy. And in fact, Schwann cells export lipids instead of lactate to the medium. And this probably is one of the bases of the leprosy neuropathy. And maybe using this model, we can find a drug that could reversible this neurotoxic effect. And this is the list of collaborators. Dr. Michele Medeiros and my PhD student Karina Girardi make most of this data together with Dr. Bruno Mieto and all these collaborators for sure. And this is a picture of my group. Arthur is not with us anymore, and Thiago, that's missing the picture, is also part of my group. So that slides, I end the presentation, and now we can make question of this third part of the presentation.
Thank you, Flávio. Very interesting talk. So, any question for Flávio?
I saw two questions from Marcos. Many receptors present in the plasma membrane have their function modulated by interaction with cholesterol. Yes, that's a point. And that's one problem to use statins in the patients. And he make a question, does hemiliprin infections change cholesterol levels in the membrane of the infected cells? Yes, hemiliprin infection alter cholesterol levels in the membrane. And does statin treatment modulate this profile? Yes, modulated. I will share again my screen. I somehow was already expecting this question, and I have this answer here. The cholesterol content increased during infection and is reduced by simvastatin. And we have in another article quantifying cholesterol. I think that not in the surface. Maybe Christina could help me with that question. I think that not in the surface. We always measure cholesterol in the cell or in the supernatant. Have you know, Christina or Angela, if some of you already measure cholesterol in the membrane? Maybe with some fluoride.
No, Flávio. Until now, we didn't measure the cholesterol in the membrane, only in the supernatant or intracellular. I have a question just out of curiosity. I saw that among these possible metabolites identified in the supernatant of the infected Schwann cells, you detect oxysterols. Do you know what kind of the oxysterols are present in the supernatant of the lactated treated?
No, unfortunately, all this identification was made by HPTLC based in the migration pattern. We don't succeed to have any data from the mass spectroscopy. I have some problem with mass spectroscopy because nothing that I try to do with mass spectroscopy work. And it's a shame because I really will love to see these carnitine acyl long chain acids in our model. That's the lipids that make all this problem in the Milbrand article. But we try it, but we just doesn't see lipids in the mass spectroscopy.
Okay, thank you, Flávio.
There's a question about ApoA. It's a protein that acts in the regulation of plasma and issue cholesterol levels in your studies. Did you verify any action of this protein in this pathway involved in the case of this neural damage?
Yeah, this is a very nice question. ApoA is involved in Parkinson, and we think about that, and we check that together with Roberta Lemes and Cristiana Macedo, and we observed that ApoA is increased in fat Schwann cells, or decreased. I don't remember anymore. Do you remember Karina? Karina was the person that made this analysis. I think that's it's decreased.
Decreased in Schwann cells.
Yeah, it's decreased. And but we stop that. We don't make any further analysis about ApoA. But ApoA is responsible to bring lipids from Schwann cells to neurons, and that's the way. But they make damage in Parkinson and in Alzheimer. It's a misfunction of ApoA, and it would be nice to observe the same occurring in leprosy, but we just doesn't put more energy on that.
By the way, I'm sorry to interrupt, but just to mention that was a comment on Science yesterday, discussing whether infection could actually be associated with beta amyloid plaques inside cells in Alzheimer's disease. So the trigger could be, and the idea is that actually those beta amyloid plaques are proteins associated with resistance can actually tackle and arrange along the bacteria, blocking the replication of the bacteria in intracellular bacteria, or viruses. So I can forward to you later, but it's very interesting because there was a $1 million award, a question on one of those foundations suggesting that Alzheimer's could be triggered by infectious disease. And so people are now trying to understand how actually those proteins that are being accumulated, and the idea is that it's a check and balance kind of thing, right? I mean, we reach a level in the elderly that, accumulation of those proteins are no longer able to be degraded. So if you have an infection after, 80, 85 years old, it's far more difficult to get rid of the leftovers of that infection, right? So it's amazing. The hypothesis is very, very nice, and it fits with the association that we are seeing also. For example, there are genetic studies, and we are also investigating it. Isabella who made the question, is one of my PhD students who is actually trying to understand a little bit of the polymorphisms that could be associated also with leprosy per se, or some sort of specific phenotype of leprosy. But there are few papers in the literature suggesting that specific haplotypes of ApoE are associated with leprosy outcome. So it is amazing how those all of those information are actually converging, and so we should.
Yeah, Parkinson and leprosy is also related.
Of course, yeah.
So they are hypothesized that these plaques are also associated with innate immune response.
Exactly. And it could be the idea of I mean, if you have a chronic infectious disease in the central nervous system, right? And for example, some of those viruses, herpes simplex viruses, for example, is one of the suggestions that we were making in this comment. Anyways, it's a very nice discussion, but it's also very interesting that whether you are and evaluating the host metabolism, looking at genetics or transcriptomics or metabolomics, we have the same pathways being detected, right? And the good thing now is that we do have all of that information. We have to put it together and trying to understand how genetics could favor one specific type of response or some sort of activation are also leading to that. I mean, we know that immunometabolism is, and these innate responses are in the basis of the outcome of the disease, right? We didn't discuss, but Roberta could also mention what we have on kino pathways and the tryptophan pathway, to be more exact, and how that also contributes towards neurotoxicity, and but it's basically the same that we are discussing for the lipids, right? But they are happening all together, and so it's really amazing how in other words, I mean, how subversion of the Schwann cell metabolism or the macrophage metabolism, reconfiguring the transcriptional pathways can make the host, permissive to the replication of the bacteria and then to the spread of the disease.
And you generated data related with microRNA doing that? Because you are pursuing that idea.
Yeah, we can. I mean, we have the data. We have to look specifically to these pathways, but I mean, we do have the data from reanalysis. This is published, recently published, part of Tiago's PhD thesis, and we have RNA-seq analysis from our skin biopsies, and this is actually will be out soon as well. But anyways, I mean, we can look at some specific pathways that are being upregulated or downregulated. And one very interesting thing that is also, you mentioned in the beginning of your talk today, is that we are seeing, and this is the part of the reanalysis of the data, we are seeing that lepromatous patients, they do have a dedifferentiation of keratinocytes in the skin, right? So this epithelial mesenchymal transition that we are seeing in Schwann cells, it's not only happening in Schwann cells, it's also happening in keratinocytes. So, and so we are favoring a kind of, activation pathway towards a more undifferentiated state of the cell that then will be shaped according to M. lepre metabolism needs. And this is again, it's very, very interesting because we don't have a lot of organisms that are able to actually induce so easily this reconfiguration in the cells, right? So it is happening in the nerve, in Schwann cells, and it's happening in the keratinocytes. So all we need is live M. lepre to induce this kind of response, right? And again, we do see a lot of upregulations of lipid metabolism pathways and lipid metabolism genes. ApoE, for example, is one of the genes that is being upregulated in the skin. So and that's the point, right? We should take the data that Karina was saying that ApoE protein is being downregulated, and so we need to compare and try to understand how to explain these this kind of responses and how it would be easier to understand the immunopathology of
Milton?
Yes.
Here, Christiana. There are some points I'd like to make about ApoE because, I mean, it's a point that we are all converging to. And we should note that it's a very important protein, not only the central nervous system about the lipid metabolism, but it's also very important in macrophages. And it's also involved in immune regulation. And we are currently working on this with macrophages, and Otto is doing his PhD on this topic. So I think we should really talk more about this topic and try to converge this data in order to, we should really converge this data to like have a more like a more broad vision of this questions, you know, about ApoE, because we have different information, and we should join this information in order to have a broader vision of this topic. And there's a point you talked about today about the statins, about this study that you're going to do on these patients in these patients with leprosy and after MDT. I think you should also check for their lipid profiles, you know, like HDL and ApoA1, because they are all regulated by this treatment with statins. And we just published some research on HDL and ApoA1. And we could talk about this later and discuss how we could do it. But
it's already there. It's already there.
Huh?
No, we are going to measure that in the follow-up. That's for sure.
Oh, okay, because
it's you know, a drug that, and I mean, it's associated with lipid metabolism. So we needed to actually even see how the levels are being are developing during the
because the statins, they will exert a direct regulation on ApoA1. So that would be very important to check, you know. And ApoA1 is also coming up, as we saw today in the morning at Academic Gallup's talk, and it's also a point that's becoming very important in our field. Sure. So, well, we can discuss this later too.
Great, great, of course.
Is everything connected?
Yeah.
I'm seeing a question from André. If there are different strains can be used in the lab, if there is any data of these different strains of imilipri and lipid metabolism regulation. It's a very nice question. First of all, Milton we need to isolate it. Milton doesn't listen to my question. Yes, André.
I'm sorry. It was cut for me. I mean, can you repeat?
Can you repeat, please? Because I think that nobody listened. Can you repeat your answer?
Okay. The question is, are there different imilipri strains that can be used in the lab with different virulence? If so, is there any data with these different strains in lipid metabolism regulation? And I was talking about the necessity. Patricia in Bauru is already maintained a group of different clinical isolates of imilipri. And I was talking with Milton of the necessity that we need to arrange a way to take a biopsy of a patient with lepromatosis, that's a different mycobacteria, to send to Patricia to try to start to make this kind of analysis in vitro, comparing lepromatosis and lipri. But at the moment, and the question is about different strains of lipri, and Thiago is my PhD student during the master thesis, use Thai hemilipri, that's is genotype 1A, and also a strain from São Paulo, that's I strain, and also a strain from Pará, that is a 4, this is a 3I from São Paulo and a 4N from Pará. And we observed that the strain 4 is, the strain 3 is, this strain may be not all the genotype 3, but this strain that we tested it's grow very slowly in the cells that we tried, and also it's pretty much more inflammatory than the 4 strain. And the macrophage in contact with the strain 3 produce more inflammatory cytokines than in contact with the strain 4 and 1. This is a data that is still there. We don't conclude that because Thiago was to a different project now, but Roberta Olmo will help me to close this data, and we'll publish that, I hope, next year, demonstrating this difference between the strains. But for sure, there are differences in the way how macrophage responds to them, but we don't make any kind of analysis in this kind of perturbation in the lipid metabolism. I believe that we will not see any difference because the signaling depends on PGL1, and how these hemilipri present PGL1. We try to analyze the abundance of PGL1 among these cells. And again, our mass spectrometric analysis that I try to do doesn't work, and we don't see nothing, any different. We saw some different, but we can't identify the ions which are different among these strains. But they present some difference in the lipid content, but we miss to identify it. I hope I answered your question.
There are questions on YouTube. So Hannah asked, sorry. Hannah said it was very interesting.
Karina, can you wait a little bit because Ximena wants to talk first. She's asking for a long time to talk.
Okay, sorry, Ximena.
It's okay, just a couple of questions. One might be, I don't know, I wasn't very good in biochemistry, so you might think it's silly. My question is, could lactate increased by exercise, anaerobic exercise, could that be helping, could it help to prevent neuropathy? And have you put these nude mice to practice anaerobic exercise to check if it does anything with this neuropathy? And the other question is regarding to the modulators that you were testing. How did you find these modulators, how they were looked for? Cortellis, integrity, what kind of platform were they looked for, or they looked in?
Modulators of what? Of lipid synthesis?
Oh, the metabolism, the pathways, the purinergic pathway, the purinergic signaling, the pentose pathways, these modulators you were testing. Well, the people at Sousa Lima were testing.
Yeah, at this moment we are testing. We are not testing. We are still trying to patronize the model. But the rationale to choose a molecule to test is based in molecules that are able to interfere with this signaling of lipid synthesis. And we know that purinergic signaling is involved in that. We also have a molecule that interfere with the pentose phosphate pathway, and some molecules that interfere in the lipid synthesis. But there are some molecules that are involved in neuroprotection that we use as control. They make neuroprotection by other mechanisms involved in just inhibit neuron death signaling, such like that. We believe that this molecule must help in our model, but maybe just use as a control. But we are open to new candidates. If you have some candidates to test, we will be glad to test it when the model is ready. Bruno is trying a more simple model in Federal Juiz de Fora, that is injecting directly in the dorsal roof ganglia and not in the nerve of the mice, but injecting imiliprid directly in the dorsal roof ganglia, and after that take out the entire roof ganglia and cultivate it in vitro. Regarding the first question about lactate and exercise, maybe Bruno could answer better than me, but I believe that there will be no modification, because Schwann cells are not very good in capture lactate from the blood. They are good to transport lactate to the axon, but not to captate from the blood. So, but also, if you increase glucose in your diet, you will not make any good at all to your axons. Giving more glucose will theoretically give more lactate to the axons, but actually glucose has all this problem that's involved with diabetes. So it's not so straightforward problem to solve.
Exercise seems a good idea.
Yeah, but exercise has lots of other effects in the immunity, so for sure will help.
Thank you.
Thank you for the question.
So on YouTube, Hannah said that was very interesting to watch your presentation, and she would like to know about this mitochondria. We know that amilopride can't induce autophagy and infected cells in macrophage, actually, and in Schwann cells was autophagic activation observed. Did you observe this process of mitophagy? Another question is, would the blockade of ROS by these lipids could induce this death of mitochondria by mitophagy?
Well, actually we observed a drop in ROS in infected cells. The infected cells present a higher glutathione response and a drop in ROS generation. Even if you came with insult from outside the cell, and we believe that the amilopride prepared that just to be able to accumulate lipids in the whole cell without have some lipotoxic effect, because lipids are very susceptible to oxidative stress. So that's why the importance to increase the oxidative response before you accumulate lipids in your house. Regarding the first question, can you repeat, Karina? I forgot the first question.
The first or the second?
No, I answered the second, but I missed
second.
the question.
Yes, I'm going to read the first question.
Mitophagy.
And yes.
I remember the autophagy and mitophagy. No, we need to do that. We have a student that analyzing, actually is a postdoc, that's analyzing the ubiquitinone profile of the infect cell. And we believe that when this data came out, we will see the entire profile of ubiquitinated proteins during the infection. And we hope that we will find PINK1 as an example. That's one of the mitochondrial proteins that became ubiquitinated before mitophagy. And we are putting our chips on that on that process. Because observe mitophagy, it's not so easy. I actually measure the number of mitochondria in the cell is also not so easy. And we believe that observing the entire ubiquitinone process, you can answer this question and also answer another questions related with the importance of arcing in the disease. But this data, I hope that will be ready along next year.
The last question on YouTube is Hannah asked if did you get to evaluate SOD or the NRF2 axis when you look at glutathione?
Can you repeat?
Did you get to evaluate SOD or the NRF2 axis when you look at glutathione?
No, I haven't.
Yes, we haven't.
It's a nice question too. I would love to make that, but I haven't. There's so many things to do and so few money and hands. You need to choose what to do, unfortunately.
So that's all. Any more questions for Flávio? Thank you very much, Flávio, for your very nice presentation.
Well, thank you, Karina. Thank you, Roberta.
Thank you, Flávio. This was
Very nice again.
I have a lot of ideas after can talk. Thank you very much.
Thank you.
Thank you, Flávio, for your contributions now and in the morning and on the first day. Thank you very much.
Okay.
Bye-bye.
Bye-bye.